To start, we are going to make a protein that is 4 amino acids long from this DNA sequence in vivo. To make this happen, the DNA first needs to be transcribed into RNA. To make this happen, we need to determine which strand is the template strand and which is the coding strand. To do this, we are going to flip the bottom strand. When we transcribe into mRNA, you will see that the T becomes a U. Now you will see that both strands do possess the most common start codon, AUG. You will also see that there is a stop codon, UAA, 4 codons down. Now that we have the coding strand, we are able to transcribe the mRNA sequence. This happens when we change all the T's in the sequence to U's. Once we have the mRNA sequence, we are able to translate it into our amino acid sequence. The reading frame starts with the start codon, AUG, which codes for methionine, then GUC, which codes for valine, AGC, which codes for serine, AAG, which codes for lysine, and then the stop codon, UAA, which does not code for an amino acid. If there were to be a mutation in the coding strand that would change the guanine of the serine to an adenine, this would also change the mRNA strand. Because of this change, it would not change the amino acid, therefore making it a silent mutation. This is because AAA and AAG both code for lysine. This is also an example of transition. If a transversion were to occur to change the same guanine of the serine amino acid to become a cytosine, this would change the amino acid sequence. AAC would code for aspirazine instead of lysine, which would result in a missense mutation. A missense mutation is changing one nucleotide, which results in the change of the amino acid sequence. Depending on the missense mutation, it may or may not affect the function of the protein based on whether or not the affected amino acid is vital to the protein function. To stick with the same codon, if a transversion were to happen in the first nucleotide, the adenine, and change to a thymine, which would make a uracil in the mRNA after transcription. While this codon used to code for lysine, after this mutation it will now code for a stop codon. This is an example of a nonsense mutation, which would make a shorter and likely non-functional protein. Now we are going to have a transversion in the stop codon. In the coding strand, the adenine has been changed to a thymine, which becomes a uracil in the mRNA transcript and makes the stop codon change into a leucine. The result of this mutation would be a read-through mutation because the stop codon would no longer perform its function and the mRNA would continue to be translated until there was another stop codon in the reading frame. This would typically result in a long non-functioning protein.